We're going to pick right up where we left off in the last lecture. This is still Chapter 7, the second half of Chapter 7, pages 176 to 191. Still linkage recombination in eukaryotic gene mapping because that's the name of the chapter, but I'm going to just basically refer to this in the syllabus and in the notes as the three-point cross lecture because that's what we're going to focus on most. So we're going to talk about three-point test crosses in our way or our strategy for mapping three linked genes on a single chromosome, determining not only their linkage order but also the recombination frequency or centimorgans or map units between them. Most of our time is going to be spent on that. But from there, we'll go on to how human genes are mapped, the different strategies used to map genes in the human chromosome. We'll talk about molecular markers. We'll define them vaguely and talk about how we map with those. And maybe just introduce a few of the more common and more modern mapping techniques very, very briefly without going into the details of those. But before any of that, let's do a review. You should be able to easily address this question given what you should have understood from the last lecture. And if this throws you, same as always. Either go back and review the old material, go back and review the old notes, refresh your mind as to what was working last time, or come see me. Come see me and we'll review it together. If you can't do this problem, you probably shouldn't move ahead. So colored something or other in the kernels of corn is due to the dominant allele R. The recessive allele, little r, when homozygous, produces colorless stuff. The plant color, not the kernel color, is controlled by another gene with two alleles, big Y and little y. The dominant Y allele is a green color in the plant. Homozygote Y allele gives you, recessive Y allele makes the plant yellow. We have a test cross between a plant of an unknown genotype and phenotype and a plant that is homozygotic recessive for both traits. The following progeny were obtained. I take one look at this. And I know the recombinant and non-recombinant progeny. I know the recombinant and non-recombinant gametes. I know the linkage pattern in the parent. That's all I need was one look. Remember, the smaller number is always the recombinant results of crossing over. So I know that colored and yellow and colorless and green are the results of recombination. So colored is big R, and yellow is little y, and colorless is little r, and green is big Y. These are the recombinants. And then I know the other patterns are the result of no recombination. So colored, and green, and colorless, and yellow are our non-recombinants. I also know that these alleles were in a coupled configuration in that initial parent because we have the dominant with the dominant and the recessive with the recessive under non-recombinant conditions. I can also determine the map distance between these genes. Now, can't I? To determine the map distance, all I need to do is sum up the total number of recombinants, which I know are the smallest ones, so that's 20, and divide it by the total number of overall offspring. So that's all of these numbers summed together. That I should have really done this ahead of time, but this is going to be 180 plus 20, so that's 200. So that's nice and easy for us. So this is going to be 10% recombination frequency. These genes are 10 centimorgans apart, 10 map units apart. Hopefully that's review, and hopefully you're okay with all that. Today we're going to build on all of those principles, and we're going to start to consider three-point crosses where we map three separate genes that are all linked on a single chromosome. We'll determine the map distance between those genes using recombination frequencies. If you remember, because I introduced it in the last lecture, these double crossover events, these three-point crosses, cause us to miss recombination of frequencies because, first of all, the genes are further apart than we're accustomed to on a single chromosome, and the second flip restores the parental configuration between the two outer genes. So we underestimate the map distances. Here's what we mean. If we have, when we start in the parent, a coupled configuration where dominant A is with dominant B and recessive A is with recessive B, the first crossover event flips the B alleles. The second crossover event restores them. So ignoring what's in the middle, if we're just looking at the relationship of the A allele with the B allele, what we see in the parent is what we get in the offspring. And so they look like non-recombinants, but in fact they are the result of two crossover events. So not only are double crossovers detectable when we do 3-point crosses, actually the double crossover events help us to instantly determine the gene order when we're doing 3-point crosses. Here's the result of a double crossover event when we're looking at 3 genes simultaneously. 3 genes simultaneously. It's the same example I showed before, but now we have a gene in the middle. We see the double crossover event restores the parental configuration with respect to A and C. But because we have a gene in the middle, we can monitor that. What I'd like you to do is look long and hard at this and tell me what you notice as the result of the double crossover event. When you compare what was in the parents with what you get in the offspring after double recombination, what you see is that the middle gene is flipped, but the outer genes are restored. So that's really the essence of gene mapping using 3-point crosses. You look for that middle gene and you spot it by looking at the gene that's flipped. The gene that I always say isn't traveling with his buddies anymore. On a 3-point cross, on a double crossover event, only the middle allele is flipped. So how do you construct genetic maps using this? If we're going to consider 3 genes, then we got to get comfortable dealing with 3 different traits at one time. So here's a practice example of that. We're going to deal with flies. We're going to talk about eye color. So scarlet eyes is recessive to red eyes. Ebony body color is recessive to gray body color. That's our second trait. And small bristles on the body is recessive to normal bristles, our third trait. So 3 separate traits. These genes are all linked, and they're all found on the fly's chromosome number 3. We're going to take a true breeding, red-eyed, gray, normal bristled fly, so that's homozygote dominant across the board, and mate it with a true breeding, scarlet-eyed, ebony, small bristled fly, recessive across the board. This is the cross we're doing. So we know the linkage pattern in terms of dominance with recessives. When we do this cross, the F1s are going to have all dominant alleles on one chromosome and all recessive alleles on the other chromosome. We know that because that's the result of this mating. When we write these genes in this way, please keep in mind the gene order is just a guess. E is not necessarily the middle gene. We just have to write them all together as linked, and so this is the only way that we can do it. We still don't know which gene is physically in the middle. So, as I said, this gives us the heterozygote, which I described. Under here, the Tegrity toolbar is the recessive chromosome from this parent, so all the recessives are here. We would call this a coupled configuration because all the alleles are of the same type on the same chromosome. We perform a test cross then. Remember, test crosses are always going to be our friends when we're doing gene mapping experiments, gene mapping problems. So here is our test cross. We have our coupled heterozygotic female, in this case, mated with a homozygotic recessive male for all 3 traits. And these test crosses are going to allow that heterozygotic parent's alleles to show through. All the crossover events, all the recombinations, everything. And since we're looking at 3 different genes all linked together, encoding 3 different traits, we will see 2 to the 3rd power classes of traits, or 8 different possible combinations or groupings of traits. Here's our cross. Here's our 8 different traits. We can have all wild type, all mutant. We can have the ebony and spineless mutant, along with the red eyes. We can have the scarlet eyes, with the normal body color and the normal bristles. Or you can look through this list, but we have every single possible combination of phenotypes. All the dominant traits, all the recessive traits, 1 dominant trait with 2 recessive, blah, 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 blah. These are our 8 classes. Sometimes in a real-world experiments, a class or 2 might be missing because the progeny number was so small you actually didn't get one. I would never do that to you on an exam. If it happens to you in some other test, say an MCAT maybe, don't let it freak you out. Just because the 8 classes are possible doesn't mean they're all represented. The same rules to the problem will apply. Take a look at this. Tell me which gene's in the middle. You can do it. Remember, the smallest number of progeny is going to represent the most infrequent rare event. What do you think is the most infrequent rare event? No recombination at all? I'll give you a hint. That's the most common. One recombination event or two recombination events simultaneously? I'll ask the question a little bit differently. What is the most rare, smallest chance of happening? Not winning the lottery next month? Winning the lottery once next month? Or winning the lottery twice next month? It's the same question. Obviously, the double recombination event is the least common. It's the most rare. So the double recombination event is going to give you the smallest number of progeny. That's 1. So we know that our double recombinants are these guys. The largest number of progeny are the non-recombinants. That's where no recombination occurred at all. The intermediate numbered progeny are the result of single recombination events. They are rarer than no recombination, but not very rare. The rarest, smallest number is the double crossover events. So we just identified our double crossovers. And we know something that we didn't know before, which is that when there is a double crossover event, the middle gene is the only one that's flipped. Or in other words, the middle gene is the only one not traveling with his friends anymore. What's the group of friends when there's no recombination? Dominant with dominant with dominant? Recessive with recessive with recessive. Remember, always ignore the bottom chromosome. That's the result of the test cross. It's not informative. These are the packs of friends. Dominant with dominant with dominant. Recessive with recessive with recessive. We know these are the double crossovers because they're the smallest number. What we need to identify now is which one of these alleles is no longer traveling with his friends up there. Well, ST and E are still traveling together. Both the dominants are together, both the recessives together. The odd man out is SS. SS is recessive here, no longer traveling with the friends it was traveling with before. SS is the gene in the middle. The highest number of progeny will always be the non-recombinants. And hopefully you get what I mean, or you're starting to see what I mean when I say that the test cross lets those heterozygotic traits show through. We ignore the bottom chromosome. The heterozygotic parents' chromosomes are what is dictating the phenotypes in the offspring. The result of double crossovers will always be the fewest progeny. The double crossover event switches the middle gene and only the middle gene. So by identifying which allele has been switched relative to the other two, you find your middle gene. It's the only one that was flipped. This is what we started with in the parent. All the dominants together, all the recessives together. This is what we get in the double crossover. This dominant's still with this dominant, but this guy wasn't here before. This guy wasn't here before. And so this is what actually happened. What actually happened was option 3. If ST was in the middle, the first crossover event would switch ST and SS relative to E. The second crossover event would switch the end genes back to what they were. The middle gene would be switched. These would be the combinations of recombinant chromosomes if ST was in the middle. We know these are the double recombinants because they're the smallest number, and this isn't the pattern we see here. If E was in the middle, we would see this pattern in the double recombinants. And we don't. We see this pattern in the double recombinants. We're confident these are the double recombinants because they're the smallest number. But these patterns don't match. The only way this works is if SS is in the middle. With SS in the middle, double recombination gives us the pattern that we are actually seeing in the progeny. And so SS is in the middle. Always look for that odd allele out, that flipped allele, the one that's no longer traveling with the other alleles it used to travel with in the non-recombinants, and that will be your middle gene. It's that easy. For 3-point crosses, the non-recombinants and the double crossovers only differ in one allele. If they differ in more than one allele, you're not looking at the double recombinants, and you've got the wrong allele in the middle. So, how do we do map distances with these 3-point crosses? We now know the correct order of the genes. We know the gene that's in the middle. Our next step is to calculate the physical map distance between them. It's the same procedure we used for 2-point crosses, except we only consider the genes 2 at a time. We're still going to take the number of recombinants over the total times 100. That wrinkle is that we have to always ignore one of the 3 alleles, one of the 3 genes. So let's do this problem, and let's consider first the distance between SS and ST. So that's the problem we're going to do. Here are the numbers we had before. We're considering the distance between SS and ST. We first, always, first step is determine the non-recombinants. These are the non-recombinants. It's the highest number. We are ignoring E. We are determining the distance between SS and ST. These are our non-recombinant configurations. The 2 dominants together and the 2 recessives together. And what we're going to do is total up anything that differs from this as a recombinant. Anything that differs from that pattern is recombinant. So here it's dominant ST with recessive SS. That's different. So we're going to count that. Here it's recessive ST with dominant SS. That's different. We don't see that up here. So that has to be the result of recombination. This is the dominant with the dominant. Well, that matches. But Dr. M, those are well, they're not recombinants with ST and SS. But recombination occurs, but not between ST and SS. We're only considering ST and SS, and this configuration matches this configuration, and so we leave them out of our total. Here the recessive ST is with the recessive SS. That matches our non-recombinants. We leave it out. Here ST dominant is with SS recessive. Well, that's different, so we'll count that. And here ST recessive is with SS dominant. That's different, and so we'll count it. So our total number of recombinant progenies are 50 plus 52 plus 5 plus 3. And that's 110. And then we're going to divide that by the total number of progeny that's here. The problem tells us that's 755. We do that, we get 14.6 as our recombination frequency or our map units. It's that easy. Can you calculate the distance between SS and E? Pause the lecture. Get out your scrap paper and give it a try. That's the only way to be sure you can do it. So same idea. If we're doing SS and E, now we're going to ignore ST. Just ignore it. Pay no attention to it. And we're going to do that comparison only of SS and E. Here it's a recessive with a recessive. That is parental. Here it's a dominant with a dominant. That is parental. Here it's recessive E with dominant SS. We don't see that pattern up here, so that counts as a recombinant. Here it's recessive SS with dominant E. That is also different, so we count it. Recessive with dominant differs. Dominant with recessive differs. And so for calculating the recombination distance between SS and E, it's 43 plus 41 plus 5 plus 3 divided by the total 755. That gives you 12.2% or 12.2 centimorgans. Finally, you can check yourself by calculating the distance between the 2 genes on the end. And to do that, we're going to ignore the SS in the middle. We're going to ignore the SS in the middle. So in this case, ST dominant is linked with recessive E. Well, we don't see that up here, so this counts. Recessive ST is linked with dominant E. That's different, so it counts. Dominant ST is linked with recessive E. That's different, so it counts. Recessive ST is linked with dominant E. We don't see that pattern up here, so it counts. And then finally, dominant is linked with dominant. That matches. That matches. But you know as well as I do that that is the result of the double recombination. And the double recombination restores that parental configuration. So we can count these if we'd like, because we know recombination occurred. And we can count these as well because recombination occurred. In addition, this total is 8. But how many recombination events occurred these 8 times? How many recombination events occurred? 16. This number 5 represents 5 times that we had a double recombination event. So 5 times these 2 recombination events occurred. So that means that's 10 total recombination events. This 3 represents 3 times that this double recombination event occurred between S and T. And since that double recombination event occurred between S and T twice for each of those 3 times, that's 6 total recombination events. We didn't have to do that before because those double recombination events only occurred between S, T, and E. So we do that total. We put it over the total number of offspring, which is 755. So it's our 50 plus our 52 plus our 43 plus our 41 plus 5 and 3 for the first set of recombinations between the two, and then 5 and 3 for the second set of recombinations between the two, giving us our total 16 because they were double recombination events, gives us 202. Put that over our total and we get 26.8. And we can take all of those numbers together and put together our physical map, just as we did in the last lecture, and come up with 14.6 centimorgans between ST and SS, 12.2 centimorgans between SS and E, and a total of 26.8 centimorgans between the two N genes, ST and E. Looks good to me. Perfect problem. On pages 182 and 183 of your textbook, the author does what I consider to be a very, very good job of giving you a step-by-step, broken down strategy for solving all of these types of problems. I almost think of it as a checklist of how to solve 3-point cross problems. If you like that kind of mental exercise, if you like having a guide, please, by all means, look at those pages. All right, now we're going to move on to concepts that are a little bit different, interference and coincidence. We're leaving the realm of recombination and talking about some of the other kind of things that happen. Everything we've discussed so far in this course well, at least for linked genes, assumes that each crossover event is completely independent of any other crossover event. That one crossover event occurring in one location has no effect or no outcome on any other crossovers occurring anywhere else. And that's just not entirely true. It's just not the way it works. Sometimes, oftentimes, one crossover event happening makes another in the same region less likely. It's almost an inhibitory effect, where we already had a crossover here and we're not about to have another. Sometimes, more rare, but it happens, one crossover event in a location makes the second crossover event more likely. And so it's almost like it primes the region of DNA for recombination in general, and so you get enhanced crossovers. We call these hotspots or hyper recombination. So most assuredly, they are not independent events. Sometimes they affect each other negatively, sometimes they affect each other positively, but they're affecting each other. Coincidence, the term coincidence, is a measure of how often two crossover events coincide or occur together at a specific region on a chromosome. And then there's a mathematical coefficient of coincidence that's used to calculate the interference, and this is the other idea, how much one crossover event suppresses another. I won't go into the math. I'm not going to tell you how to calculate these. I just want you to know that these exist and what they mean. Coincidence measures how one crossover event makes another more likely. Interference sounds like what it is. Measures how one crossover event makes another less likely. So what's the effect of multiple crossovers? We've only discussed these classic examples of crossovers between the two inner chromatids of a homologous pair during synapsis, the kind of typical, straightforward, true crossover, called a crossover and shown with an X because of the configuration that the chromosomes take. But that's not all that's possible. Crossing over is possible between any two chromatids of the entire tetrad. So you can get two-strand double crossover, that's the double crossover we've already talked about. You can get a double crossover that involves 3 strands. So one strand, one chromatid, is involved in 2 crossover events, but with 2 different chromosomes. You can get 4-stranded double crossovers, where you have 2 chromatids in the middle crossing over, and the 2 outer chromatids are also crossing over with each other, and that becomes crazy too. You can get mixes and matches of all these different kinds. Now, what does this do for genetic recombination, genetic variability? It throws it through the roof. It means parental chromosomes are being shuffled like crazy prior to the formation of gametes and fertilization. Remember, that's what we wanted. Remember we talked about being on a hot planet? It's really, really hot all the time, but then it gets really, really cold, and you want to be able to adapt. This is making that adaptation possible. By introducing genetic variation into the species, you make some individuals more fit than others. But as geneticists, this is a nightmare. This makes identifying recombinants and non-recombinants extremely difficult. Usually, as a result, as the physical distance between genes gets bigger, so too does the inaccuracy of the estimated genetic distances in map units. So we can see that. Here's the actual distance between genes, and here's the recombination frequency or map unit between genes. Early on, when the genes are close together, well, that was awful. Early on, when the genes are close together, it's very, very linear. One does a good job of measuring the other. But if this was to continue to be a linear line, we would see that correlation carry true. But it doesn't. We can see that that starts to curve that line. And the correlation between the recombination frequency that's measured and the true actual distance between the genes diverges, because this number is no longer accurate, and this number is the true measure. So we have to be mindful of that. It's not a perfect system. So let's close this lecture talking about how we map genes in general, both low-tech and high-tech, both old-fashioned and new way, especially as it pertains to the human genome. We've said it before when we talked about pedigrees and things, but we can't do controlled crosses with people. People have this insistence on all these confounds that make experimental studies with humans quite difficult. So when it comes to mapping genes in the human genome and determining linkage patterns for human genes, we need to go beyond this idea of controlled crosses, test crosses, etc. Who remembers the simple genetic tool that allows us to track the inheritance of traits across generations of a single family? I even just said it a minute ago. That's what pedigrees are. Pedigrees give us the familial information across many different generations with a decent N, because we're looking at so many people, so that we can actually see not only the inheritance patterns of traits, but even linkage patterns in humans. This technique was experimentally used for real in a real situation to determine that the gene for blood type, the ABO blood type, was physically linked to a gene that caused a syndrome called Nail-Patella syndrome. This syndrome is real, it's rare, but it's real, and it causes fingernails to either grow abnormally or be absent at all, and individuals also have missing kneecaps. They don't have kneecaps. It's rare, but it's autosomal dominant, and this is a pedigree that shows both the transmission of the blood type as well as the Nail-Patella syndrome, because they are linked. So I want you to look at these parents. One parent has the A blood type allele linked to the recessive normal allele for Nail-Patella syndrome. The other parent has, and on the other chromosome, has the O blood type allele linked to the dominant disease-causing Nail-Patella syndrome allele. The other parent is normal, has no Nail-Patella syndrome, so they're homozygotic recessive for the N gene. They carry one allele for the B blood type, and the other allele is for the O blood type. Now we can watch the transmission of these chromosomes in the pedigree, because for the blood type, the alleles always show through due to the codominance, and the syndrome is dominant. Nail-Patella syndrome. So anybody who doesn't show the syndrome is known to be homozygotic recessive. So we can look at each of these individuals in this pedigree and assign to them which chromosome they got from which parent. So what I just did there was I took the chromosome from the father and assigned it in the offspring. And these individuals had a lot of kids. They had, 8 kids. So pretty good N. And then that means the other chromosome came from the mother, and we can assign the chromosome to her too. So we can see which chromosome each of these kids got from each parent. These are not independently assorting genes. We do not see every possible combination of phenotype. These traits travel together because the genes are linked. Now some of these kids mated and had children of their own. We can assign new colors to their spouses. And we can assign also the transmission of those spousal chromosomes to their kids, as I've done here. And then what we see is one of these sons got the parental, blue, O blood type nail patella syndrome phenotype chromosome from his father. And the other kid got this chromosome. Got the O allele linked to the recessive normal nail patella allele. And that chromosome is not present in any of the members of this family. Now we know that chromosome didn't come from the mother, because the mother gave the top chromosome with the A allele and the normal allele. So this chromosome had to have come from the father, but the father doesn't have this chromosome. So where did this chromosome come from? This chromosome came from a recombination event in the father during gametogenesis, during spermatogenesis. The recombination event flipped the N alleles relative to the I, so now the normal N allele came down to be on the same chromosome as the O allele here. Recombination event. We can do the same for this mating. Let's assign Brown to the wife in this case. We can do the same assigning. The wife gave her recessive O, recessive normal nail patella allele to all of her children. The father gave these alleles to his two children, but then this allele pops up. Same thing. An allele, a chromosome that is not present in the father, but attributed to the father due to a crossover event that flipped those N alleles. Amazing! Amazing to witness recombination in the human genome by looking at nothing more than a family tree. Just amazing. So that's one way to map. By doing that type of analysis with pedigrees, we did mapping. We show conclusively that the blood type gene and the nail patella gene are on the same chromosome. That's the only possible explanation for this data. But what are some other ways we can map with the human genome? We're well into the age of molecular genetics and molecular technology, so we don't need to rely solely on physical observable traits for genetic studies, nor do we need to rely on invasive or familial studies for genetics. We can now look directly at the DNA in an almost non-invasive way. I mean, if you consider wiping the inside of your cheek with a Q-tip as invasive, then this is invasive technology. But if you've got no problem taking a Q-tip and rubbing the inside of your cheek with it, we can get DNA from that, plenty of DNA from that, and do genetic studies that way. Molecular markers are deviations in DNA sequences that are known to be highly different between individuals in the human population. But they're not really mutations because they're not occurring in genes, so they have no effect on the individual at all. It's not as though the marker reduces fitness or can be observed on someone's face or skin. This is only a DNA deviation. Some of them are referred to as SNPs, SNPs, which stands for single nucleotide polymorphisms. This is where a single DNA base pair, which in this case should be a cytosine base pair to a guanine, is something different, like a thymine base pair to an adenine. A single nucleotide change or a single nucleotide polymorphism. That's one type of deviation, one type of molecular marker. We also have what we used to call RFLPs, restriction fragment length polymorphisms. I won't go into what those are, but they're often the result of SNPs as well. And we have what are referred to as microsatellites. We talked about microsatellites already, believe it or not, in our very first lecture. These are the repeats that cause Huntington's disease. Very, very rarely you find these in real genes, and they can cause diseases when they go awry. Most of the time, these short stretches of repeating DNA sequence are found in non-coding genes, which have nothing to do at all with coding proteins. And so whether you have many, many repeats or a small number of repeats, it doesn't affect the individual because that DNA is not doing anything. So these are all molecular markers. These are all, again, places where people differ from one another, and it doesn't cause disease, and it doesn't cause a problem, and it's completely benign. Those details are not critical right now, although for those of you who are forensics majors, it's the cornerstone of forensic DNA technology. What's important for us now in this lecture is that each of these molecular markers are very easily detected using current technology. They're inherited because, of course, the DNA came from our parents, and they can, and often do, lie near or are linked to disease-causing genes. And so what happens then is we have an easily measured, easily detected marker, easy to see. You can think of it as easy to see. right next to a disease-causing gene that is critically important for human health, but not easy to measure and not easy to see. So what we do experimentally is we track the marker. And by knowing that the marker is linked to our disease-causing gene, we are indirectly tracking our disease-causing gene, although we are only measuring the marker. So we use this kind of approach to map important genes by seeing how closely linked they are to these unimportant but easily tracked markers. The analogy I use for this is the idea of pizza directions. I was raised in Brooklyn, so we would always use this joke of pizza directions. Pizza directions is that you never tell somebody that, go 5 miles on this road and make a left onto L Avenue, Avenue L. Instead, you would say, go a bunch of blocks, and then you're going to see Jimmy's Pizzeria on the left and make that right at Jimmy's Pizzeria. And then 2 blocks later, there's going to be a bagel shop. Make a right at the bagel shop. We'd call those pizza directions. You would use landmarks in the neighborhood to tell people how to find things. That's exactly what these molecular markers are. They're pizza directions. The disease-causing gene is this house on the block. But this house is hard to see from the intersection, and this house is hard to measure experimentally. But this building right here is the molecular marker, and with its bright yellow awning, and the fact that it's on the corner, this is an easy marker to detect and an easy marker to use for your pizza directions. So if you know that the gene of interest is on the same block as your marker, if you know that the gene of interest is on the same physical human chromosome as your molecular marker, you track the presence of the gene by measuring the presence of the marker. It's as simple as that. And so that's how things are done now. And like I said, it's also how forensic DNA technology is done. It is all about the linkage pattern. It's all about a low recombination frequency between the two because they are physically very close to one another on a single chromosome. There are even more advanced mapping methods. Now, these are awesome, mind-blowing mapping methods, but all of them, no matter how advanced, how technologically revolutionary they are, all of them exist to do one thing and one thing only: show linkage, show 2 DNA sequences on the same molecule, show 2 buildings on the same block. They don't tell us the overall location. They certainly don't tell us which chromosome these genes are on. They only tell us where those genes are relative to one another. The even more modern mapping techniques give us information on location as well. These are called physical mapping methods instead of genetic mapping methods. This is where I know you're 17,000 base pairs away from the end on chromosome 17. And deletion mapping techniques exploit that relationship. Deletion mapping allows us to get chromosomal information as to the location of genes. The logic of these is simple. You get a large number of deletions on a single chromosome, and you compare the presence and absence of mutations on those different deletions. Very simply put, there's a deletion mutation on chromosome 5. That same cell is now missing your gene of interest. Guess where your gene of interest is? There's a big deletion on chromosome 5, and your gene is gone. Where's your gene? Well, it was somewhere on that deletion on chromosome 5. That's why it's gone. The logic is as if a bomb hits Main Street. Your house was just destroyed by a bomb. Guess what house guess what block your house is on? It's on Main Street. So, how do you do these deletion maps? Well, we use test crosses. You get an individual that is recessive or mutant for the entire set of traits you're looking for. You also get a large collection of deletions of individuals with different deletion mutations on their chromosome. And by comparing the presence and absence of mutant alleles in the presence of absence of the deletions, you can map the order and location of the genes you're interested in. Okay, so this technique was used to map the gene that causes Duchenne muscular dystrophy. And here's how it works. This is what we're going to do. You have your collection of deletions. In this case, the line for each deletion mutant represents which part of the chromosome is missing. So all of the rest that we don't see is a part of the chromosome that's still there. So this part of the chromosome for deletion number 4 is still present. The line represents what's missing. And down here is where the mutation for each of these genes is present. M is where the mutation is present, and where that mutation is masked because the chromosome isn't deleted in that region for each of these different genes. So we're going to use this information to order these genes on the chromosome. And here's how we're going to do it. At each beginning or end of any of these deletions, we're going to draw a vertical line down, such as this. Vertical line down, vertical line down, vertical line down, vertical line down, and then here, and here, and here, and here, and here, and here. So I just drew vertical lines down at all of the beginnings and ends of these deletion lines. And then what we're going to do is come to our map here, and we're going to ask these types of questions. This region here of the chromosome, this region right here, only deletion 1 has that region missing. All of the other deletion mutants have that region of the chromosome present. So let me get rid of some of this mess. So we're asking ourselves, are there any mutants that are showing the mutant phenotype only for deletion 1? Here's deletion 1. Are there any mutants that are showing only the mutation for deletion 1 and not for any other? So here's mutant B. Mutant B shows its mutation in deletion 1. And for none of the other mutations do we see that mutant B. That means that the B gene is present on a part of the chromosome that is missing in deletion 1 and only deletion 1. Well, that part of the chromosome is right here. There's the only part of the chromosome that's missing in deletion 1 and only in deletion 1 is right here. So gene B is first. Now we move to our next bin, our next region. And for this, we're going to look for a gene that is mutant in both deletion 1 and 2, but no other. Is there any gene down here that shows its mutant trait in the deletion 1 and 2, but no other mutants? So, deletion 1, deletion 2, mutant, and no other. Gene C shows the mutant trait in deletion 1 and deletion 2, but in none of the others. So that means gene C must be here. Moving on. Is there any gene, I'm just going here because it's the largest space, it's going to be easier to see. Is there any gene that is showing its mutant trait in deletion 2, 3, 4, and 6, but no others? 2, 3, 4, and 6, but no others. They say no, there aren't. Are there any genes that are showing their mutant trait in deletion 3, 4, and 6 only, and no others? 3, 4, and 6 and only and no others. Well, that's E. 3, 4, and 6 and no others. So E must live here, because only this region is missing in deletions 3, 4, and 6 and no others. Since that deletion is present there, the mutation in E can't be masked. Any deletions showing their any mutation showing their mutation in 4, 5, and 6 and no others. 4, 5, and 6 and no others. 4, 5, and 6 and no others. So that means A must live here. A can't live here, because if A lived here, we would not see the mutation in deletion 5. Deletion 5 contains DNA in this region. For the mutant A, its mutant phenotype is showing through even in deletion 5, so we have to look for the region that has 4, 5, and 6 all deleted. What are we missing here? F. Gene F exists in a region that is missing in 5 and 6, and 5 and 6 only. The only part of this chromosome that has 5 and 6 and only 5 and 6 missing is here. So this is where gene F lives. Gene G is missing or shows its mutation phenotype only in region 5. Well, where is only deletion 5 missing and no other deletions missing the chromosome in that region. That's here. G. So I've got A, B, C. So D is what I'm missing. So D is only present or not masked in the deletion 5. So now we're looking for a region that's missing in all of the chromosomes except deletion 5. 1 is missing here, 2 is missing here, 3 is missing here, 4 is missing here, 6 is missing here, 5 has DNA in this region, and so gene D must live here. And so using deletion mapping, we can map the order of the genes. The order of the genes on this chromosome is B, C, D, E, A, F, G. Phew. Even these methods that I'm presenting here are fairly obsolete. We now have technology that's almost like science fiction, where we can physically manipulate DNA and see where particular genes are with our aided microscopic eye. This is called in situ hybridization, and there's other related techniques, but what you're looking at here are fluorescently labeled tags that are physically binding to specific genes at their chromosomal location and then glowing unique colors. So based on the way the experiment is set up, we know that the green tag is identifying one gene we're interested in, the pink tag is identifying another gene, so we can see, physically see with our eye where these genes are and what chromosomes they are on. With results like these, you know exactly where your gene lies. You don't need to genetically map, you have physically mapped. You've spotted the gene with a tag. Recombination rates vary widely between species. They even vary between different types within a single species. The different strains that scientists use exhibit different recombination rates. Different genders within a specific and single species often show different recombination events. Humans have twice as much recombination as mice and rats. We're not really sure why that is, but it is. Human chromosomes number 21 and 22 have the highest levels of crossing over, and they are the smallest chromosomes, counterintuitive. Chromosomes 2 and 4 have the least amount of recombination, although they are not the largest chromosomes. Female autosomal chromosomes undergo 50% more recombination than the very same autosomal chromosomes in males. Not quite sure why that is. 60% of all human recombination occurs at a fairly limited number of what we refer to as recombination hotspots. So the vast, vast majority of recombination in human cells occurs over a fairly small swath of DNA regions. Where those hotspots are differ between species, even between us and our closest genetic cousins, chimpanzees. Hotspots of genetic recombination are different. And for almost all of the observations on this slide, no one has the slightest clue why. So what did we talk about in this lecture? Well, we talked about double crossovers causing us to miss recombination events because they restore the parental pattern. That causes us to underestimate map distances in 2-point crosses, but that's okay because we have 3-point crosses to track all that. In all of these cases, we're doing our test crosses to let that heterozygotic parental pattern show through. In all cases, the highest number of progeny will always be the non-recombinants. The double recombinants will always be the fewest number of progeny. Once you identify the double recombinants, because they are the fewest number, you look for that odd man out, that flipped allele, the allele that's no longer traveling with its friends, and you will have found that middle gene. We define coincidence as a mathematical value that measures how often two separate crossover events will coincide or occur together. Interference is how much one crossover event will suppress another. And then we talked about mapping. As the physical distance between genes grow, so too does the accuracy, the inaccuracy of the estimated genetic distance. So then we've got to go to other techniques. We can use pedigrees that can show us, sometimes using familial information and inheritance patterns, how different genes are linked to one another. I'm sorry, the LOD scores I cut out of the lecture, so don't worry about LOD scores. We can use deletion mapping. Deletion mapping exploits these naturally occurring chromosomal deletion mutations. We can get a big collection of them together and use that to physically map genes. We went through that problem together. And then we ended the lecture talking about things that affect recombination rates, even though we don't know why. Gender affects it, species affects it, DNA sequence or chromosomal location affects it, and we really don't understand why. So we're going to switch gears completely now and move on to some of the large-scale chromosomal changes that can occur that really funk up the chromosome molecule itself. And are technically mutations, chromosomal mutations. I don't want to confuse that with the other type of mutations we'll cover later on in the semester. But we're going to talk about the chromosome as a molecule in the next lecture, and what can go wrong with that molecule and how that affects the way that genes interact with each other. So we can kind of put to bed the transmission genetic stuff for a while, and we're going to switch gears and talk about some of the other DNA-related processes as they affect genetics more on the molecular side than on the transmission or Mendelian side. As always, thanks for watching, and we'll cover this material in class.